Dear students, I'm Dr. Nidhi Sharma, p.g.govt.college, Jhalawar Associate Professor, Zoology. In this part of the muscular system, I shall tell you something about the excitation and contraction coupling of a muscle fiber. Here you can see in this case the nerve ending, motor nerve ending, which has been cut open, and you can see the axon terminal, the synaptic vesicles, the mitochondria, and the synaptic cleft. The synaptic vesicles contain acetylcholine neurotransmitter, which transmits the nerve impulse along with the help of the calcium ions. Now you can see the sarcolemma. The black arrows of which release the acetylcholine into the cleft continue near under the sarcolemma. Here you net in the first stage, net entry of sodium ions initiates an action potential which is propagated along the sacrolemma and down the T tubules. The T tubules, along the T tubules, the sodium ions are released and the action potential are propagated. The SR tubule has been cut open. The SR tubules are purple in color. In the second step, action tubule potential in T tubule activates voltage sensitive receptors, which in turn trigger calcium release from terminal cisternae of sarcoplasmic reticulum SR into cytosol. Here you can see the calcium ions are released into the cytosol of the muscle cell. In the third step, calcium ions bind to troponin. Troponin changes shape, removes the blocking action of tropomyosin. Actin active sites are then exposed. In the fourth step, which is the contraction, myosin heads depicted by the orange globules alternately attach to actin and detach, pulling the actin filaments towards the center of the sarcomere. Release of energy by ATP hydrolysis powers the cycling process. Here, when the calcium ions are released from the actin filament by active transport into the sarcoplasmic reticulum after the action potential ends. In the sixth stage, tropomyosin blockage is restored, blocking myosin binding sites on actin, contraction ends, and muscle fiber relaxes. The muscle relaxation mechanism can be said that the first acetylcholine esterase, an enzyme present in the neuromuscular junction, destroys acetylcholine, preventing continual stimulation of the muscle fiber. Then, calcium ions are transported from the sarcoplasm back into the plasm, plasm reticulum SR. Third, linkages between myosin and actin are broken. Fourth, then the muscle fiber relaxes. So, this was the muscle relaxation mechanism. Now, if we talk about the various bands and zones, you can see in the photograph, the first photograph shows a relaxed muscle, the middle shows the size is shortening, and the last figure shows that the contraction has taken place and the muscle has shortened. In this case, in contraction, A band stays the same. I band gets smaller, H zone gets smaller, sarcomere shortens, and contraction takes place. This is again the pictorial depiction of the sliding filament model of contraction. In the first figure, you can see the size, the longer size of the muscle. Second figure shows a little contraction has taken place, and the third figure shows the highly contracted muscle filament. Now, where does the energy for contraction come from? Muscle cells require huge amounts of ATP energy to power contraction. The cells have only a very small store of ATP. There are three different pathways that supply ATP to power muscle contraction. ATP is initially supplied from first cellular respiration. If ATP is abundant, it is converted to creatine phosphate and stored in skeletal muscles. When ATP is low, creatine phosphate supplies phosphate to ADP, making ATP. Creatine phosphate and adenosine triphosphate only stores remain good for about 10 seconds for a maximum contraction. ATP must then come from cellular respiration or glycolysis. Here in this diagram, you can see the methods of regenerating ATP during muscle activity. In the first case, the energy source is creatine phosphate. Here you can see the creatine phosphate gives out creatine and one inorganic phosphate, and ADP diphosphate changes to ATP. This is direct phosphorylation coupled reaction of creatine phosphate and ADP. The energy source in this case is CP or creatine phosphate. Here oxygen use is none. Products are 1 ATP per 1 creatine phosphate. Duration of energy provision is just 15 seconds. In the second instance, glucose, when glycogen breaks down or is delivered from blood by the process of glycolysis in cytosol creates ATP. 2 ATP net gain takes place. This is again changed to pyruvic acid, which changes to lactic acid, and here no oxygen is required as shown in the figure. This is an anaerobic mechanism. Glycolysis and lactic acid formation takes place by anaerobic method. Energy source here is again glucose. Oxygen use is none. Products are 2 ATP per glucose molecule. Lactic acid and duration of energy provision is 30 to 60 seconds. In the third case, glucose from glycogen breakdown or delivered from blood is changed into pyruvic acid. Then fatty acid, amino acids, as well as pyruvic acids in the mitochondria as shown in the diagram with the help of aerobic respiration give out carbon dioxide, water and ATP, 38 units of ATP per glucose molecule. Here this reaction is aerobic mechanism and is also called aerobic cellular respiration. The energy source is glucose, pyruvic acid, free fatty acids from adipose tissues, amino acids from protein catabolism. Oxygen use is required. Products are 38 ATP per glucose, carbon dioxide and water. Duration of energy provision is for many hours. This, students, finishes the muscular system which I have taught to you in great detail. Thank you.